Okay, in this section, I'm going to answer some questions related to osmosis and diffusion. And this first aspect is the multiple choice question, which is section A. So you choose option A to D and you choose the best answer from each question. So the first one, the movement of particles from a region of higher concentration to a region of lower concentration until equilibrium is reached is known as osmosis, diffusion, active transport, and plasmolysis. So that's a clear definition of diffusion. Okay, so if you watch my videos, you'll be able to get a clear understanding of that. So, which of the following is essential for osmosis to occur? Okay, so osmosis require what? A selectively permeable membrane. Very important for osmosis. Okay, during osmosis, the particles that move across the selectively permeable membrane are, so we have solute, we have solvent. So the particle that pass through it is water molecules. Water molecules actually pass through it, not the solutes, because the solute dissolve into the solvent, and water molecules actually pass through the membrane. Okay, a plant cell placed in a concentrated sugar solution will most likely, so a sugar solution is a strong solution. So what happens? It becomes what? Plasmolyzed. Okay, it becomes plasmolyzed means water is going to be since it's a strong solution, water will tend to leave the cells and become what? Plasmolyzed. Then which of the following factors increases the rate of diffusion? A. Decrease in temperature, decrease in concentration gradient, increase in surface area, increase in distance over which diffusion occurs. So the clear answer is increase in surface area. This helps diffusion to occur easily. So a red blood cell is placed in distilled water. What is most likely to occur? So when you put it in distilled water, you know what happens? It becomes what? Turgid. And what happens? It may actually burst. So it becomes turgid and may burst. That is what haemolysis. The movement of water molecules from a region or higher water potential to a region of lower water potential through a selectively permeable membrane is known as, a membrane is there, is known as what? Your osmosis. So which of the following structures in plants is mainly responsible for absorbing water from the soil? So that is your root hair, not the stomach, not the gut cell, is your root hair. And don't forget, absorbing water from the soil is a process of osmosis. A plant cell is placed in a dilute solution, that is your normal water. Water enters the cell and the cell becomes, once it enters the cell, what happens? The cell gets filled with water and expands. So use the term we use there, we use the term turgid. Become turgid. Become turgid. Which of the following is not required for simple diffusion? Okay, ATP energy, it's not required. Okay, because this does not require energy. So ATP energy is not actually required for this experiment. A student places a drop of ink into a beaker of water. After some time, the ink spread throughout the water. This demonstrates what? We did something on before, when you add the ink, everything starts spreading. So the good example of diffusion. Which of the following would occur when a plant cell is placed in a concentrated salt solution? So it's straightforward. Cytoplasm pulls away from the cell wall because it becomes plasmolyzed. The shrinking of the cytoplasm away from the cell wall in a plant cell due to loss of water is called plasmolysis. So those get used to those terms, very important. Which combination will result in the fast rate of diffusion? So your high temperature makes diffusion to occur very fast and large surface area. Which of the following is an example of diffusion in humans? So movement of oxygen from the alveoli into the blood in respiration. We're taking oxygenated blood. The main the main difference between diffusion and osmosis is that osmosis involves water movement through a selectively permeable membrane. So this selectively permeable membrane makes osmosis to stand out. A cell has a higher concentration of sugar inside it than outside. If the surrounding membrane is permeable to water, but not sugar, what will happen? So if the membrane is permeable to water and water can easily enter inside without sugar, what happened to the cells? The cells, the water will what enter into the what is moving into the cells, and when it moves into the cells is much, the cell become what very turgid. Which of the following will reduce the rate of diffusion? So, increase in diffusion distance. I explained this in my video about this diffusion. The increase in the diffusion distance, because it will take the molecules a longer time before it can easily was spread out to all part of the container or all part of the surface area. When a red blood cell is placed in a higher concentrated salt solution, it will what? So if you add a red blood cell in a highly concentrated salt solution, what happens? It shrinks. It will shrink because water is going to what escape from the cells and it shrinks. Which of the following is an importance of osmosis in plants? So it helps the root hairs to absorb water from the soil. So you absorb the water to the soil into all other part of the plant for it to what to perform properly and to be able to live properly. Okay, so that is that. So now I'm going to go through some theory questions. So the first one is define the following terms. So the first one is diffusion. So and this is the movement of particles from a region of higher concentration to a region of lower concentration. Osmosis, require a membrane. So movement of water molecules from a region of higher water potential to a lower water potential through a selectively permeable membrane. And what is a selectively permeable membrane? So a membrane that allows some substances to enter through it. Allows some substances to pass through it. That is what a selectively permeable membrane is. And what happens is actually prevent others from passing through it. Okay. So state three difference between diffusion and osmosis. So diffusion can involve solutes or gas particles. Osmosis involve only water molecules. In diffusion, it may occur without a membrane, while osmosis requires a membrane. In diffusion, it occurs in gas and liquid, while osmosis occurs in solutions. So give two examples of diffusion in living organisms. So we have oxygen moving from the alveoli into the blood, and carbon dioxide moving from the blood into the alveoli. And you give two examples of osmosis in living organisms. Water absorption by the root hairs and movement of water into plant cells. So it's a very good example. Okay. So now we have a table here. Now a student carried out an experiment using potato cylinders. Equal size piece of potato were placed in solution of different concentration and left for 1 hour. So we have solution A, B, C, D, and E. And this is the initial mass. So the initial mass is the mass of the potato before it was placed in the solution. So you have to record the mass before you put it into the solution. Then the final mass is the mass you obtain after you leave that slice of potato into the solution for 1 hour. That's why you can see that it actually tends to increase in mass, but some of them tend to decrease in mass. For example, if you look at solution D, initial mass was 5.0 gram and the final mass was 4.3. The same thing for initial mass for solution E. So it decreases in what mass, which means something have occurred. For it to decrease, it means water actually left the cell. The cell have made the potato actually made the size of the potato to reduce, so the mass is going to reduce. So if you can use your ideology for plasmolysis and hemolysis, or for turgidity and flaccidity, you'll be able to understand that okay, one of them was placed in a strong solution that made water to leave the cell, or one of them was placed in a hypotonic solution that made water to fill into the cell and increase the mass of the cell. You understand? So this gives you a initial ideology. So with this, I can tell you that okay, for solution A, this is 5.0, which was the initial mass, and it increased in mass, which means it was placed in the solution in a hypotonic solution because water will tend to enter into the cell and make it to increase in mass. The same thing for solution B, and the same thing for solution C remains unchanged, which means solution C was placed in a hypotonic. in the isotonic solution, which is what in neutral solution, which means there's no change in mass, it remains the way it is. But for D and E, if you look at it, it has reduced in mass, which means it was placed in a hypertonic solution, just for you to understand what this table is all about. So the question is, calculate the percentage change in mass of potato in solution A and B. So if you check your calculation here, you can see, so solution A, so 6.2, which is the final mass minus the initial mass. So 6.2 minus 5.0 divided by the initial mass, which is 5.0 times what? 100. So it give you what? 24% increase. So it has actually increased in size. But for solution D, which is this one, I explained that water was being reduced because it was placed in a strong solution. So if you do some same calculation, 4.3 with the final mass minus the initial mass, which is 5.0, divided by the initial mass, which is 5.0 times 100, it gives you a 40% what decrease. But if you look at it, it actually decreases in size, which is what 4.3 here. So it gives you what 40% decrease. Okay, so which solution appears to have a concentration closest to that of the potato cell sap? So the answer is C. You understand? C is the closest to the concentration of the potato cell sap. And why? Because if you look at that C, if you look at C here, if you look at this, the mass remains what? Unchanged. It remains unchanged. 5.0 initial mass and 5.0 final mass. So there was no change in mass, indicating no net movement of water. So that's the answer. So state what happened to the potato in cell A. So cell A, the potato cell gained water, it gained more water and become what? Turgid. So it increased what? In mass. While for solution E, the potato cells lose water and become less turgid and become flaccid, which means plasmolysis actually occur because water has been what? Escaped from the cells. So if you look at this table, for example, if you go back again, for A, you can see A increased in mass to 6.0 gram. For E, solution E, you can see it reduced in mass to 3.8 gram. So that's it. So state 3 precautions that should be taken to ensure reliable results in this experiment. So the first one, use equal size potato cylinder. Keep the volume and the concentration of the solution constant and accurately measured. So the one I did in my last video, when I did in the kitchen, and actually I didn't actually do the main measurements, but I was able to put them in a well organized level. So I was able to put both solutions in almost the same number of volume. Okay. So the next one, the potato pieces have to be dry. You have to dry them before taking your final mass. So after you remove them from the solution, you dry them and you take the final mass to get an accurate reading. Explain why a red blood cell placed in distilled water may burst. So once the red blood cell takes in water by osmosis, it swells and may burst because it has no cell wall. Okay, it had no cell wall. So if you take too much amount of water, it becomes very turgid, like that of a balloon. Then what happens? It bursts. What will happen to the same red blood cell if it were placed in a highly concentrated salt solution? So once you add a strong solution to it, what happens? Water will tend to leave the cell and it will lose water and shrinks. Okay. Explain the process responsible for the change in A and B. So the change occurred by osmosis due to differences in water concentration across the cell membrane. So you give the biological term used to describe the bursting of a red blood cells. That is what? Haemolysis. That's called what? Haemolysis. So question 4. A plant cell is placed in a concentrated sugar solution, which is also a strong solution. Describe the movement of water molecule in this situation. So water will move out of the plant cells into a concentrated sugar solution by osmosis. So the water will leave the cell and go into the sugar solution due to the process of what? Osmosis. Instead of entering into the cell, the water will leave the cell and it becomes flaccid and plasmolyzed. Describe the changes that occur in the cell. So the cell loses water and the vacuole becomes smaller and the cytoplasm pulls away from the cell wall. Okay. So name the process responsible for these changes. That is what? Plasmolysis. Explain how the cell could be returned to its original state. So you put this back into a hypertonic solution. So the cell can be returned to its original state by placing it in a dilute solution of water, allowing water to enter into the cell by osmosis. And this is called the plasmolysis. So once we put it back into water, it gives it some time. What happens? The cell will become what? Turgid. State two factors that affect the rate of osmosis. So differences in water potential and concentration gradients and the temperature tend to affect the rate of osmosis. So if you understand all these questions and you're able to answer all these questions properly, trust me, there's nothing in cell and its environment. There's no question that comes from cell and its environment that you won't be able to answer. Okay, so see you in my next video and thank you.